This is the top 1000 previous year question series for CSIR NET Life Sciences, and in which 10 to 30 questions are discussed today. Okay, let's go. Here is the first question, guys. Rudkin hypothesis is related to first option A. Mating in ham of polygamous male. B. Elimination by deleterious mutation by sexual selection. Mate selection process by female. Evolutionary arm race between host and parasite, which is the correct option. D is the correct option. The evolutionary arm race between host and parasite. Rudkin hypothesis related to the host parasite interaction, competitive interaction, and the predator prey interaction, and such interactions, the coevolution, etc. So the Rudkin hypothesis is related to the evolutionary arm race between host and parasite interaction. This is the correct answer. Next question. In pre-industrial period in England, prepared moths had light coloration which effectively camouflaged them against light colored trees and lichens. During industrial revolution, many lichens died out and trees became blackened by soot from factories, and interestingly dark colored moths were predominantly seen. This happens due to natural selection of dark colored moths when first option, natural selection of dark colored moths which were initially present in fewer numbers. Okay. Second option, new mutation which arose due to the environmental pollution. Third option, macroevolution occurring due to the environmental change. Fourth option, natural selection of camouflaging mechanism of moths. Is it correct option? No, there is no camouflaging mechanism. And third option is completely macroevolution. It is a wrong option. And second option, there is new mutation which arose due to environmental pollution. But the very correct answer is the natural selection of dark color moths which were initially present in fewer number. Natural selection is the process here. Due to the industrial revolution, there is purple moths had light coloration which effectively camouflaged them against light colored trees and lichen. When industrial revolution, there is trees become blackened, so the natural selection, there is also the moths are also becomes dark colored in their feathers or their wings. Okay, this is actually happen. There is a natural selection process. Next question. Wolbachia are obligate intracellular bacteria. Many different strains of which are abundantly present in insects. They induce mating incompatibility in hosts. Males infected with one strain can only fertilize females infected with the same strain. No other pathological effects are observed in hosts. A possible evolutionary consequences of this phenomenon would be: first option, the extinction of many insect species. Second option, termination of sexual reproduction in many insect species. Co-extinction of host and parasite. Reproductive isolation leading to rapid speciation in insects. The correct option is fourth option. There is reproductive isolation leading to rapid speciation in insects. This is the correct option. What is they ask here? The Wolbachia, obligate intracellular bacteria, have their males are infected with one strain, can only fertilize females infected with this same strain. Okay, there is an reproductive isolation takes place here. Due to this, there is a new speciation or rapid speciation in insects also takes place. Okay. Males are only fertilized with the females infected with this same strain of this bacteria. Okay, that's what happens here. So the correct answer is reproductive isolation leading to the rapid speciation in insects. Next question. There is a question of Hardy-Weinberg principle. Frequencies of alleles A as capital A in a population at Hardy-Weinberg equilibrium are 0.7 and 0.3 respectively. In a random samples of 250 individuals taken from the population, how many are expected to be heterozygous? This is the questions. So we want to calculate this Hardy-Weinberg equation. For the Hardy-Weinberg equilibrium, what is the equation? P square plus 2pq is equal to q square is equal to 1 is the Hardy-Weinberg equation. Frequency of allele A is here 0.7, that is 70% of allele A, small letter A, that means A we already taken it and seen that small letter A is 0.3, that is 30%. Okay, then we get p square 2 plus 2pq plus q square is equal to 250. We have to find out the 2pq heterozygous condition here. So 2pq is equal to 2 into 0.3 into 0.7 is equal to 4.2 0.42, and that is 42%. So 42% of 250 is equal to 105. So heterozygous females are 105 or 105 heterozygous conditions are there. That's what they asked in this question. Okay, next question. What is the frequency of heterozygote in that is AA in a randomly mating population in the frequency of dominant phenotype, that is dominant phenotype, that is 0.19. Okay, what that's the question. The equation is Hardy-Weinberg equation, p square plus 2pq plus q square is equal to 1, and p square plus 2pq is equal to that is they are we want to consider q also, because 2pq also, because there is in condition, so we want to consider the p also, 2pq also. Then we get the p square plus 2pq, what is there is they given that 0.19, and q square is equal to 1 minus so q square is equal to 1 minus 0.11, and q is equal to 0.9. We get q is equal to 0.9. We know p plus q is equal to 1. Okay, and p plus q is equal to 1. So p is equal to 1 minus 0.9, that is 0.1. 0.1 is the answer. So the heterozygous is 2 into 0.1 into 0.9. We get q is equal to 0.9 and p is equal to 0.1. So heterozygous condition A is equal to 2pq is equal to 2 into 0.1 into 0.9 is equal to 0.18. Next question. Tay-Sachs is an autosomal disorder. The frequency of affected newborn infant is in 14,000. Assuming random mating, what is the frequency of Tay-Sachs is an autosomal disorder. The frequency of affected newborn infants is about 14,000. Assumes the random mating, what is the frequency of heterozygotes? This is the question. That means frequency of affected newborn is 14,000. Assuming random mating, what is the frequency of? We want to calculate the heterozygous conditions. It is an autosomal disorder. So the autosomal disorder, what is the affected newborn means there is small aa, that is the affected newborn condition. So q square is equal to 1 that is this q square is equal to 1 pi 14,000, that is 0.000714. We get the answer 0.000714, and q square we get 0.085 is in when we calculate this is q square is equal to 0.085, and we already know that p plus q is equal to 1. Then we get p is equal to 1 minus q, that is 0.9915. okay, then we get the frequency of heterozygote, that is 2 into pq, that is 0.17. we get the answer 0.17 here. Okay, that is q square is equal to, q square means the frequency of dominant recessive condition q square 1 by 14,000, that is the affected individual. Okay, then we get the heterozygous condition 2pq is equal to 2 into 0.9915, and heterozygous condition, when we calculate the heterozygous condition we will calculate 2pq. okay, then next question, the following genotypes were observed in a population. HH is 90 and heterozygous HH is 60, and small HH is 50. Which one of the following is correctly frequency of H allele, that is dominant H allele, and what will be the expected number of HH in the given population? Okay, these are the conditions, options. First option, okay, we want to calculate these conditions. We want to calculate expected number of HH in the given population. Okay, so HH we number of individuals is 19, heterozygous HS condition is 16, and small hs is 15. So frequency is frequency we will get 0.45, 3 and 2.5. when we'll calculate 90 by 200, that is total number of individuals. By 90 by 200 is equal to 0.45 total number of individual. 60 by 200, that is 0.3, that is the HH condition, and 50 by 200 0.25. So the frequency of H allele is what is 0.45 plus half into 0.3. okay, but why the half condition? Because dominant condition we want, so half 0.3. here we use the half of 0.3, 1 by 2 of 0.3. so we get 0.45 plus 0.15 get 0.60. the answer is 0.60 frequency of H allele. okay, then frequency of H allele in this population, small letter allele in this population is, there's some clarity, is half 1 by 2.3 is equal to 0.40 is the frequency of H allele in this population. okay, so according to Hardy-Weinberg, p square plus 2pq is equal plus q square is equal to 6.6 whole square, 2 into 0.6 into 0.4. 0.4 is the q, that is small h, and plus 0.4 the whole square, we get the answer, that is 72% of capital small 72% of capital H condition, capital H dominant condition in this population, that is 200 into 36 by 100, we get the 72 capital H, percentage of capital H in this condition. Okay, next question is, what is genetic drift? Random and directional change in the allele frequency simply as a result of change from one generation to next in the small population is called a genetic drift. Sudden or by chance change in the population is called the genetic drift. It is only for the small population, counted for the small population. Large population we cannot calculate the genetic change. Okay, so random and directional change in the allele frequency as a result of change from one generation to the next for a small population is called the genetic drift. Okay, we want to solve the genetic drift problems. What is bottleneck effect? Bottleneck occurs when there is a disaster that reduces the population, which rarely represents the actual genetic makeup of a initial population. This leaves a small variation in the surviving individuals. Bottleneck effect is affected due to these disastrous conditions, or some earthquake, or some other flood, etc. In this condition, bottleneck effect is represent the actual genetic makeup of the initial population. This leaves small variation in the surviving individuals. Surviving individuals has a small variation in the actual genetic makeup. Okay, this may occur due to the environmental disaster of species or habitat destruction. Then the founder's effect, loss of genetic variation that occur when the new population is established by small number of individuals from original population. This happens, founder's effect is happened due to some migration process. When a group of rabbits migrated to one population to the other side, or between the a sea or a river. Okay, they migrated to the one side of this river to the other. Then there is some loss of genetic variation occur there. Okay, this is the founder's effect. Then we have to solve these problems of genetic drift. According to genetic drift, bottleneck and founder's effect, they tend to the dash the gene variation among the new population. Options are increases, decreases, stabilize, remains same. According to genetic drift, bottleneck effect and the founder's effect, they tend to the decrease in the genetic variation. Decreases, this is the decrease, there is decrease in the gene variation among new population. Decreases the gene variation in the among the new population. Next question. Next question. Northern elephant had seals the reduced to 20 individuals in 18,000. Biologists studied the variation in proteins in this species. They found no genetic differences in the protein among different individuals. This lack of variation is due to the fact that elephant seals live in constant environment where there is no need for the gene variation. Population bottleneck neck and genetic drift, natural selection resulting in the single best genotype, a very low rate of mutation. They said the here you can see 20 individuals, only 20 individuals, they consider 20 individuals, so small number of population. So bottleneck effect is genetic drift occurs for a very small number of population. Okay, so we can consider eliminate these options: very low rate of mutation, natural selection resulting in a single genotype, and fact elephant seals constant environment where there is no need of gene variation. So the correct answer is population bottleneck and genetic drift. They consider, the biologists consider variation in proteins of species, okay, for a 20 individuals. So we can go through the answer, that is the population bottleneck and genetic drift. Next question: 20 small populations of a species, each polymorphic for given locus, were bred in captivity. In 10 of them, the population size was kept constant by random removal of individuals, while the other 10 were allowed to increase their population size. After several generations, it was observed that in 7 of the size restricted populations, only T was present. In the remaining 3, only small t was present. In the growing populations, 8 retained their polymorphism and 2 only small t was observed. The experimental illustrate the genetic drift was, which is more likely to in larger population. Genetic drift which is more likely in small population. Density dependent selection against T and density dependent selection against T. Genetic drift which is likely in large population, genetic drift which is more likely in small population, density dependent selection against T, density dependent selection against T. So we can eliminate the last options, density dependent selection against T and density dependent selection against T, small t. So there is no need of these type of answers. We get genetic drift more likely in large populations. It is never genetic drift in small population. Genetic drift only occurs in small populations. So we consider these we consider genetic drift which is more likely in small populations. Okay, next question: which of the following is true of genetic drift? It can decreases the genetic diversity in a population, it can increases the genetic diversity in a population, it plays much larger role in determining the genetic makeup of original population, it results in a new population having all allele which were present in the original population. So the correct answer is we know the wrong options are, we want the true of the genetic drift. So it can increases the genetic diversity in the population? No, never. It can decreases the genetic diversity actually. So B option is eliminated. It plays a much larger role in determining the genetic makeup of original population. So it results in new population having all the allele which were present in the original population. It is not a true option. So very true option is it can decreases the genetic diversity in a population. Genetic drift is decreases the genetic diversity actually. So the correct answer is it decreases the genetic diversity in a population. So the correct answer is the first option is the correct option. So thank you guys, thank you very much, and please share and subscribe for my channel. Okay, thank you.